So the first part of chapter 11 was about DNA and DNA replication. So you have to have DNA, you need to be able to replicate it to pass it on to new cells. You replicate it during synthesis stage of the cell cycle. But then once you get to that G1 and that G2 stage, you have to carry out the basic things you need in order to carry on life. That comes down to gene expression. DNA has the information that will allow life processes to occur. Gene expression is how we take that DNA, turn it into an RNA molecule, and then produce a protein. It's these proteins that will make the phenotypes that we see on individuals. So we were talking about, if you have a bent or a straight pinky. Well, whether or not you have that is whether or not you have a gene that tells you to lay down the bone in a certain way so that it either grows straight out or it has a little ding in it. So it's the proteins that get laid down or the enzymes that are made that will make something like bone that will determine your phenotype. So this is how we go from the genotype to the actual phenotypes we express. So this first video is what is called the central dogma. How we go from DNA to RNA to protein is called the central dogma of biology. And so this is an overview of the two processes that we're going to do in the last two assignments before the exam. The first part of the central dogma is called transcription. So if you transcribe something, you're rewriting it. So remember, DNA and RNA are both nucleic acids, but we're going to take it from one language or from one type of molecule into another. We're going to rewrite DNA in the letters of RNA. So to transcribe means to write. So we're going to rewrite. So remember, the DNA is in the nucleus. So all of this is taking place in the nucleoplasm of the nucleus. And this is why we rewrite it. Remember, we put the DNA behind a nuclear envelope and a plasma membrane. We put it behind two protective barriers because it's so important. Well, when we make a protein, that's going to happen in the cytoplasm, not as protected. Remember, that's outside the nuclear envelope, only has one protective barrier. So we want to keep this really safe, and we don't need all of it at one time. So we're just going to rewrite a little bit of it. So transcription, we're going to start with DNA, and we're only going to open up a little bit of it. Remember, we have a double-stranded. We're only interested in a little bit of it. So the gene is what we're going to transcribe. And we're going to take that DNA and we'll make it into an RNA molecule. So this will be a copy of the gene. Now, when we get done with making that copy, this is just called a transcribed gene, or it can also be known as a primary mRNA molecule. So this is going to be the messenger. Remember, this is a copy. So remember, we had those three types of RNA. So this will be the mRNA. This will just be, like say, a little bit of it. But the second part of transcription, so first we're going to make this primary mRNA, and then we're going to process the primary mRNA, and it will become a mature mRNA. You can think of this as, if a writer writes a script for a movie, well, they put down what they think should be in the movie. But then you make the movie, and not necessarily everything that went into the script ends up in the movie. So first we're going to write out exactly what we see here, but then we have to modify it a little bit, and so that is going to be part of the transcription process. All of this is taking place in the nucleus plasm. This mature mRNA is what leaves the nucleus. So that is transcription, the first half of the central dogma. After we have our mature mRNA, we can do the second part, and that is translation. So we now have an RNA molecule that has just left the nucleus. We now need to make a protein. So this is when we're going to take the mature mRNA and we will actually make a protein out of it. So this is called translation because we went from macromolecule language to protein language. Remember, proteins are going to be made up of amino acids. So this is a collection of nucleotides. We're now going to translate that into a series of amino acids. So translation is just like if I went from England to France. Transcription is like rewriting it. America, if you're an American and you speak English, so do the English, but it's not exactly the same. They have different words. So we call the trunk of a car a trunk. They call it the boot. But it's the same basic language. But then if you were to go from England to France, now you're in a whole different language. You can think of the nucleic acid language as English and proteins as French to help you remember that translation comes after. So this is where we will find a ribosome. This mature mRNA will have to find a ribosome, which remember is made up of an RNA molecule itself, that rRNA. So we will then use RNA in order to translate our mRNA, and we will make a polypeptide. So we will go from a series of nucleotides. So here is a series of nucleotides, and now all of a sudden we will have a series of amino acids.